We're going to move on now to hear from Professor Albert Tenesa, who's personal chair in Quantitative Genetics, it actually shared between the Roslin and the MRC-Human Genetics Unit in the University of Edinburgh. Albert is a global authority in the application of quantitative genetics as it pertains to disease manifestation. And his fundamental work over the last number of years has been to try and understand phenotypic differences across individuals in a population, which is in essence what many of us as clinicians are trying to do. Am I choosing the right medicine at the right time for with the least likelihood of harm? And it's an absolute pleasure, Albert, to have you joining us this afternoon with CORONAGENES, and goodness knows we could do with help there. So Albert, over to you, sir. Thank you.
Thank you very much. Okay, so I'll have a slightly different perspective to the previous speakers, but my aim is basically trying to engage in discussions about how we do science and talk about what we've done with CORONAGENES and how we actually took advantage of SHARE. I am a huge fan of SHARE. I think it's fantastic. It worked extremely well for us. It's not expensive at all. So I only have good things to say about SHARE. Anyway, I'll move on. So what is CORONAGENES? Can't seem to move the slides. Okay. All right. So there it is. Can you hear me? Yeah. Okay. So this is the outline of my presentation. So the background to the projects, what we aim to do, what we had to do, our launch, some stuff about IT and security that we had to go through, the questionnaire, some of the results, and some of the conclusions and my own thoughts. Okay, so this started at the heart of the pandemic on 21st of March. Jim Wilson and I had a few text exchange, and we thought, what can we do with the pandemic? Can we actually reuse some of Jim's group work on Vikings 2? So Jim had been recruiting online, had a system to recruit online through Vikings 2. And since they couldn't recruit because all other non-COVID research had been stopped, we thought maybe we could reuse the infrastructure and the staff he had for his project. The good thing about recruiting online is that it's cost effective. You don't need research clinics, and you can do this with relatively few funds. Actually, we did this without any formal funding. We reused funding that we had, which has good things, but also bad things. It also increased we also thought that we could increase the speed of genotyping, and I'll say why by using direct-to-consumer genotypes from volunteers. And direct-to-consumer genotypes, for those that don't know, are basically you can buy a DNA kit on the web, and they send you a saliva kit usually, and then you send the saliva kit, and they send you the results back of your DNA, basically genotype for one of the standard genotyping arrays that we use for genotype. The advantage of that is also that you could potentially recruit volunteers worldwide, and also be inclusive in terms of recruiting people from different ethnic backgrounds. And you can do that all through email and social media, contacts through the press. Okay. So why direct-to-consumer genotyping? So these are data. It's a bit old, but it kind of shows how big the market is. So these are two of the main direct-to-consumer genotyping companies, and it is estimated that between the two of them, they probably have about 25 million, well, and the others, which are very minor, about 25 million clients. These are basically people that pay for their own genotyping, the same genotyping that we would have to cost in a research grant. So what were the objectives? The objectives were to recruit tens of thousands of volunteers online with and without symptoms of COVID. We were more interested on the symptoms of COVID, that whether they had COVID or not. And the reason for that were that there were already ongoing studies on COVID, mainly on people that were on clinical care, like that led by Kenny Bailey. So we consented people broadly for future health studies. We asked them to recruit, to record their phenotypes, the baseline data, and then when they develop COVID, daily symptoms, and then follow-up data. So we wanted also to consent people to be able to sample them for DNA, because we imagined that we would get some people that would share their genotype results, but not everybody. We also wanted to do a fingerprint antibody test, do global recruitment, but more importantly, recruit people that we could link to electronic health record in the UK. So the three main kind of research questions were what are the genetic influence to COVID, and the symptoms, and the severity. Can we predict severity? Can we predict who is going to end up in hospital from the early symptoms and the genotype? And then we wanted to study outcomes from post-infection. So who is going to develop post-COVID, or can we predict who's going to develop long COVID from the genotype? In terms of how we did it, we had to go pretty quick. In early March, we had the idea. We submitted in April the paperwork to a court at the University of Edinburgh, and then on the 30th of April we got court approval, and then we submitted for ethics, which also took about 3 weeks. And when we had the approvals, we actually launched on the 15th of June. And about 2 weeks later, we approached share participants by sending them about 125,000 emails. We were quite slow for our taste, I guess, at that point with a court sponsorship. They were worried about two main things: holding of such large number of personal data and data security. They were mainly concerned about people uploading their own genotypes to our systems. So obviously, we went through ethics. We went through penetration testing. That was a requirement from a court. And penetration testing basically is linking with a company that tries to hack into your system, and if they manage, they tell you what is wrong, and if they don't manage, or they see things that could be improved, they basically tell you what to do. One of the main worries that the penetration company was actually people getting into the system from within the university. So if a student a student account is compromised, it suddenly becomes a lot more easier to compromise into any university system. So I don't know. We basically gave them a user so that they could try to do it within our systems. And then we had to set up some kind of data validation and cleansing. We had to make sure that the genotypes data that people submitted were not just worthless data. In terms of that, so we had an initial landing area where we quarantined the files, and we did some basic inspection of the data and rejected the file or not. Then we had a versioning system. So if someone submitted 2 different files, we had to record what file came first and what file came second. So and we had to know that it was from the same person. Then we had to look those files for protection against our system, code injection, and kind of general garbage that might be in the file. So basically trying to avoid that people put nasty things into the university systems. And then we had to do standard virus scanning. So this is kind of the process that participants follow. The participant hears about us through a number of systems. They register their interest. They get an automatic ID. They get an invitation. They might ask some questions, and then they consent and complete the questionnaires, and that's transmitted to the database. So that's basically how the registration of interest looks like. It's relatively simple, and that's how the system works. So you have different keys where you can answer the baseline questionnaire, your daily questionnaire, and a link to how to upload your genotype file. And these are the number of recruitments that we've had so far. So we have about 20,000 people recruited to the study. And I think this graphs clearly shows the benefit of working with share. So this was the number of participants the first 2 weeks, basically. That's how well we could do through social media and newspaper. But when we sent invites to share participants, as you can see, the numbers jump quite steeply. I think, our success, our recruitment rate was about 10%, I would say, something like that. So I think that's quite good. And we've kept recruiting people, but obviously it has flattened quite a lot. And some of the things we've learned, I guess, in terms of recruiting, I think that there is an important question that kind of bothers me is how is it possible that there is 30 million people almost that pay for their genotyping. They give phenotypic information to these companies. These companies basically have the right to sell their data without no strings attached. How it is so difficult that for us to actually recruit people and for them to donate their DNAs, okay, or their DNA data. And my hypothesis is that these companies are very good at interacting with participants, and we scientists have this in a very one-way direction. We ask them for a lot of questions, but they get very little in return. And I think by not engaging on this kind of two-way interaction, we will be missing quite a lot of people that would participate and work with us. The other thing that I think it's important, and I think previous speakers have, Anna has talked about it, I think we need to convert biological data into data that makes us be very dynamic on how quickly we can ask questions. And I think a good example of that is also UK Biobank. If the data is available, you can answer questions a lot faster. And especially in difficult circumstances, it is a lot easier to manage data than to manage samples. So I would be a big fan of share being genotyped widely, and that's why I asked the question before. All right, so some of our results. So we did a genome-wide association study. So we basically compare symptomatic cases to controls to try to identify genes that were linked to disease. We did the standard quality control, we did the association, and then we replicated. And those are the numbers that we got. So these are the number of people that donated their DNA results. The majority of people are from Ancestry DNA, but we had to process each of these data sources in a different way because the genotypes, the SNPs between the different platforms don't overlap. So we had to process them independently and then merge them. In total, we had only 2,016 COVID cases, and the rest had no developed symptoms, so we treated them as controls. In terms of ancestry, here in blue, you can see the coronagene participants, and they clearly overlap on this PCA with samples from European descent. So we have some people of different ethnic backgrounds, but very few actually indeed, only 24. And this is the GWAS. We found only suggestive evidence of significant associations, but when we tried to replicate on one of the largest cohorts on COVID, we didn't replicate any of the findings, but we contributed data to the cohort. All right, so what are the conclusions? So we didn't find any significant association with COVID. There was no replication, so we contributed data to AGI, which reported 13 new loci. This wouldn't have been possible without SHARE. And I think direct-to-consumer genotypings have the potential to increase sample sizes on this type of genetic epidemiological studies, but we really need to understand what drives the behavior of people that have those genotypes. We need to engage with them in a more useful way, I guess. And finally, acknowledgements to Jim Wilson, that Shona Kerr, and many other people that help us, and Vasily Rapsis, who did an MSc with me over the last summer and did the GWAS work. Thank you very much.
Albert, thank you for an absolutely fascinating study